Hello guys and welcome back to Excelsior Academy. So we'll be continuing our topic of membrane solute transport. So yesterday we talked about the facilitated diffusion, right? So here also we are going to talk about the facilitated diffusion. Okay. So here we are going to discuss with the ions, okay, the diffusion of the ions by the channel proteins. So of course these channel proteins are hollow membrane bound proteins with hydrophilic gateway. Now in the plasma membrane these channels have to be hollow, correct? Because see, again they are just nothing but made up of alpha helices, right? And here it has to be hydrophilic in nature. Why? Because here we are talking about the ions. So ions, either positive charge or negative charge, they both are going to need to interact with the amino acids which is present over here. Okay, and it is also required in order to select the correct ions. ऐसा नहीं है कि Cl minus का channel है, so Na plus can come and pass through. It is not. As I mentioned earlier also, it is going to be very specific. Okay. Now, so what are the features of these ion channels? They are selective for the ions, regionally located at specific site on a cell surface, like sodium channel at the soma of neurons, functionally unique. Of course, right? Because they have to be unique. Na plus को Na plus का ही channel करेगा, Cl minus को Cl minus ही करेगा. Otherwise, there won't be any proper regulation of opening and closing of these ion channels. Okay, now ion channel selectivity depends upon the charge of the ions, whether it is negative or positive, and the size of the ion. So for example, let us say that this is K plus, okay? And if you remember, the resting potential of the membrane is minus 70 millivolt. Right, now let's talk about the K plus channel. So here if you see, this is K plus channel. So what do we see here? Okay, so this is suppose your K plus ion channel. Okay, so it is of course having the negative charge. So when I say that the channel is having negative charge, it means what? The amino acids which is present over here, okay, they are going to be negatively charged so that they can interact with the plus ions effectively, and then it will help in the selection. ठीक है? Now अभी यहाँ पे एक और चीज क्या ध्यान देना है? The size of the ion. So now you tell me, the size of K plus ion is bigger or the size of Na plus ion is bigger? कौन सा बड़ा है in the size? K plus ion or Na plus ion? For this you should know the atomic number. तो क्या है? What is the answer? Akshaya? Excuse me. Yes. So what is the atomic number of potassium and Na plus? Yes. Okay. So atomic number of K plus is 19 and Na plus का 11 है. ठीक है? अभी suppose हमने यहाँ पे एक electron lose कर दिया है, because of which it becomes K plus ion and Na plus ion. Now tell me, which of the ion is going to have bigger size?
K plus.
Correct. So it is going to have the K plus ion. Now, अब K plus यहाँ से pass हो रहा है, so it is having, so the channel is designed in such a way that it is going to accommodate only your K plus, right? अभी by chance यहाँ पे अगर Na plus आ जाएगा, so what will happen? It will be loose, right? Here the cavity will be more for Na plus. Na plus छोटा है और जगह बहुत बड़ी है. So what will happen? Because of the space is more, right, between the amino acid ka negative charge and the Na plus, there won't be any interactions, correct? So the channel will not get open because Na plus is there. So what will happen after some time? The Na plus, if suppose it is coming, it will just get out, right? So whenever a correct K plus is coming, then only it can pass through. Now let's imagine that this channel is of Na plus. ठीक है? अब Na plus की जगह पे अगर आपका K plus आ रहा है, so what will happen? The size is big over here as compared to your Na plus. So because of the size, it won't be able to pass through the channel. Am I getting? Is it clear?
Yes, ma'am.
I have a question.
Yes.
Since the channel is, can you go back to the slide before?
Yes, yes.
If you look at the channel, it looks like it is strongly negative. So it should still be able to hold on to sodium, right? Even if it's a potassium channel, simply because it's so negative.
I didn't get your question. Repeat.
Sodium has one, sodium is just like positive enough. Like it only has one extra electron, right?
Huh.
But then this one looks like it's strongly negative compared to sodium. So it should still be able to hold on, right? Simply because it's more overpowering.
Acha, don't go directly to the figure of the potassium channel actually. ऐसा नहीं है. In reality, this is just for the explanation. In reality, if this much big is your potassium channel, the only helices which is forming the selectivity filter are having the negative charge amino acid. Okay, this portion of your channel cannot have charge if you understand properly, because here it is hydrophobic. I cannot have any philic amino acid. तो यहाँ पे आपका normal hydrophobic amino acid है. It's just that here where the selectivity filter is present, which decides whether K plus should come or Na plus should come, there only the negatively charged amino acids are present. The figure is
There are
little
still
misleading.
multiple, right? There are still multiple negative
Okay.
amino acids. So
Okay, I'm coming to that. See, now look it over here. So this is a size which is allowing, okay? And kaisa hota hai? At a time, if the binding pocket is only one, okay, in the channel, let us say that this much is a space given for K plus, and this is the space for your Na plus channel. Okay, so what is happening then? When it is going, so K plus आपका freely ऐसे rotate नहीं करता है, move नहीं करता है. It is always with the water associated. कैसे होगा? If this is a K plus, okay, O minus H ऐसा. This is how the water is associated with your K plus. Getting? This is how your water hydration is present. And when it goes inside the channel, so these water molecules get removed, and यहाँ पे जो plus है, यहाँ पे जो minus रहेगा वो आपका amino acid का minus आ जाएगा. So plus minus का interaction होता है. ठीक है? Now this is happening only with one K plus, the interaction, because the space, the binding space is only for one K plus. You think, I mean, it looks like the plasma membrane is here, this much big channel is there, so many K plus can go together, but no, it is one at a time. So space ही इतना है कि सिर्फ एक ही K plus जाएगा पहले, it will interact with the other amino acids, then it gets confirmed कि हाँ यही सही है, then only the conformational change happens where the other side of the channel, like in आपका cytoplasm का side of the channel gets open up. Now are you getting?
yes, ma'am.
Sir, see, the figure was little misleading. It's just that K plus channel is having negative charge because K plus के साथ interact करना है. But the space provided in the channel is only meant for one at a time. Okay?
No, yeah, I got that. But since sodium is smaller and there are negative, there are negative amino acids meant for potassium, right? So sodium should be able to fit in and not be able to go out as well because it is bound, even though it's loose.
So that's correct. But as I said, it is having the water hydration. It is surrounded by water molecules, right? So the water molecules, when it is removed, amino acids तो आपके move नहीं होंगे, right? So when it should have that capacity to remove the water molecules which are present around the Na plus, remove and make that plus minus interaction. अभी gap इतना रहेगा, एक plus यहाँ एक minus यहाँ पे है, then you can't have the interaction, correct?
Yeah, got
it has to be at a proper distance. So that's why it is not able, it is loose for Na plus, and that's why it goes out. Now it's clear, right?
Yes.
Okay. So then we have got different types of channel. Passive, which is always open, also known as leakage channel. So this is also important that every time we have certain leakage in the channel. Now, why it is important? Can you guess? So active means gated channels. जब जरूरत है, जब impulse आता है, then only it gets open, then only it gets closed. But sometimes we also do need leakage channels. Why do you think we need leakage channels? थोड़ा बहुत तो लीकेज चाहिए ही हमें. Why?
To maintain the equilibrium.
Correct. Absolutely correct. To maintain the equilibrium. All of a sudden, suppose some signal is coming and then it goes. So आपने वो एक signal को दूसरे में pass नहीं किया है, but that particular cell got the signal. Now it needs certain time to get it normalized, right? So for that, already certain ions will be present outside or inside. So that will help in getting the equilibrium fast. Okay. अगर impulse अगर आप pass कर रहे हो, then of course you have got the proper coordination of all the channels, like gated channels are there, and we have the proper coordination of those. Okay. Now let's talk about these channels slowly. So it will be clear to you all then. Now, voltage gated ion channels. So these are regulated by the electrical membrane potential near the channel. Mainly when we talk about the voltage gated, so we are talking here about the neurons. So जो आपके brain में ये होते हैं, ठीक है? So voltage gated channels are found on the axon hillock, all along the unmyelinated axons and the nodes of Ranvier in the myelinated axons. So I hope you know all these things. तो ये आपका nerve cell हो गया है, neuron हो गया है, okay? Then you have got the axon helix जहाँ पे ये आपका bind करता है the starting point. Okay, then here we have got the dendrites also. अगर आपने figure देखा है nerve cell का, so we have ऐसे dendrites which makes connection with the another one, right? Okay. So we find all these voltage gated channels in the unmyelinated axons. Now why in the unmyelinated axons? Unmyelinated क्या होता है? एक होता है जहाँ पे आपके माइलिन शीट्स प्रेजेंट होते हैं, करेक्ट? इफ यू रिमेम्बर ऐसे है ना. Now this particular example says about the unmyelinated axons and the nodes of Ranvier in the myelinated axons. So what is happening here when you have the unmyelination? What is the speed of the impulse transmission then, Shruti? Is it fast as compared to the myelinated or unmyelinated?
Unmyelinated is slower.
Are you sure?
Yeah.
Why do you think?
Because actually myelin causes the channels to be blocked or not be present, which means that the signal will jump like throughout over the myelin, not over the myelin, as in it will jump faster, and that is why myelinated axons are generally faster compared to non-myelinated ones.
First you said unmyelinated, right?
I said unmyelinated ones are slower.
Acha, okay. Yes, correct. So unmyelinated ones are slower because then they have to complete the entire distance. In the case of the myelinated, they are going to jump from node to node, right? Okay, good. So that much is clear. So what you can see over here is that in this case, okay, where these are present, you can see that the ligand gated channels are present. Now, why it is ligand gated, we'll come to that. Then here at this end, we have got the voltage gated calcium channels. Whatever impulse is present from one nerve to another nerve, the response is passed with the help of the calcium channels. ये याद रखो, ठीक है? अब यहाँ पे आपके पास Na plus K plus channels रहेंगे in the myelin sheaths. Okay. Now, okay, look at this diagram. So this will be more clear to you. So right now, this is going to give you a picturization between a presynaptic and the postsynaptic neuron. Okay. Now, you have got the Na plus K plus Na plus K plus pump. The star ones are the voltage gated and the circle ones are the chemically gated. So chemically gated मतलब क्या होता है? With the help of the exocytosis, the neurotransmitters will be released from here over here. ठीक है? This particular region is known as synapse. ये हमने पढ़ाई है. तो ये हो जाता है प्री सिनेप्टिक क्लेफ्ट, ये हो जाता है आपका पोस्ट सिनेप्टिक क्लेफ्ट. ये हो गया सिनेप्स. सिनेप्स में क्या होगा, एक्सोसाइटोसिस से न्यूरोट्रांसमीटर रिलीज होंगे. दोस न्यूरोट्रांसमीटर फॉर एग्जांपल एसिटिलकोलिन, so they go and bind to their receptors, okay, and whatever coordinated with the Na plus or K plus or chloride channel, they will get opened up. ये एक हो गया. Now voltage gated कैसे? So they are present over here. Can you see? As mentioned, they are present in this region. And then the channels for the diffusion. So when you say diffusion channel, इसका मतलब क्या हुआ? Just we have discussed yesterday. It is based on the gradient. Okay, Na plus ज्यादा कहाँ है, K plus ज्यादा कहाँ है. So they are just simply the diffusion channel. So they'll try to attain the equilibrium. Okay, थोड़ा सा idea मिला. So slowly we'll come to know. Once we start learning about all these channels, then the entire picturization we will get it clear. अभी इतना clear है about the synapse and then how the voltage gated channels are present here, and then once the signal is released in the form of the acetylcholine, that will carry the information and bind to the chemical gated channels. And from here the signals are passed, then again comes over here, and then again the signal is passed from one neuron to the another neuron. ठीक है? Okay, now let's talk about the sodium ion channels. So when the Na plus voltage gated channel opens, membrane potential goes from -70 to less negative channels. अब ऐसा क्यों हो रहा है? Already we have mentioned that resting potential कितना है हमारा? -70 millivolt. Now what we are saying is that if Na plus channel gets open up, the -70 is becoming more negative. Let us say minus millivolt. Sorry. Okay. Now, why? Because this is because a positive ion is moving inward, making the inside of the membrane more positive. Oh, sorry, it is becoming less negative. Na? Sorry. So it is becoming -50, let us say. So यहाँ पे क्या हुआ? -70 millivolt का आपका resting potential है. Now, when the Na plus voltage gated channel gets open up, so what is happening? The positive ions are coming inside. So जब negative है और positivity increase हो रही, so what is happening? The negativity is getting decreased. Okay, here the example is given that from -70 millivolt, it is becoming +20 millivolt. Is this much is clear? Just forget about this. This much is clear, right? Na plus जितना ज्यादा आएगा, उतना ज्यादा आपका membrane potential बढ़ जाएगा. Correct? Akshay, clear? Yeah, okay.
Yes.
Now, this is how your sodium ion channels look. Okay, तो इसको हमने क्या किया है, चार में डिवाइड किया हुआ है, but eventually you have to understand that ये ऐसे अलग चार नहीं है, they are clustered together. Okay, but each domain, so 4 domains are there, each domain is having the, how many helices are there? 6 helices. Can you see? It is present, right? So यहाँ पे भी देखो 1 2 3 4 5 6 1 2 3 4 5 6 like that, you have got 6 helices in each domain, and this total together it is going to form your sodium ion channel. Okay, now look at over here, the same diagram is given. Okay, the same thing is represented over here. This is the real structure. Okay, domain 1 2 3 and 4, right? Now same thing you have to visualize over here. So this is how in reality in the plasma membrane your sodium channel looks like. Clear? Itna clear hai? Now we need to talk about the different loops which are present over here, because that is responsible for the functioning of your Na plus. Okay, let's go slowly. So the sodium channel consists of large alpha subunits that associate with proteins such as beta subunits. Okay, an alpha subunit form the core of the channel and it is a functional on its own. तो यहाँ पे alpha subunit कौन सा हुआ आपका? If you remember, okay, I'll show you in the next figure. I'll show you. So the alpha subunit has got the 4 repeat domains labeled 1 to 4, and each containing 6 membrane spanning segments labeled 1 to 6. ये clear है? So these are your alpha subunits. Okay. These are your alpha subunits. Okay. And this entire thing is your alpha subunit. Okay. It has got 4 domains. 1 to 4. Each domain has got 6 alpha helices. Okay, this alpha, ये alpha अलग अलग है, confusion नहीं होना चाहिए. So your sodium channel is made up of alpha subunit and beta subunit. This alpha subunit is made up of domain 1 to 4. Each domain is made up of 6 alpha helices. इतना clear है? Yes?
Yes, ma'am.
Okay. I'll just go to the back figure. Now, in this figure, can you see the fourth one they have shown as a positively charged? Okay. So this one acts as the sensor for the voltage. So that is your channel's voltage sensor. So just remember segment 4 is acting as the sensor of the channel. Now, voltage sensitivity of this channel is due to the positive amino acids which is located at every third position. Okay. Now, when it is stimulated by a change in the transmembrane voltage, this segment moves forward and the extracellular side of the cell membrane, allowing the channel to become permeable to ions. So what is happening? When stimulated, इसका मतलब क्या? जैसे ही आपका membrane potential change हो रहा है. Okay. So that change in the membrane voltage is being understood by this Na plus channel. Okay. So when there is a change in the membrane potential, it is allowing this channel to become permeable to the ions. Now here which ions? It is the Na plus. Now ions are conducted through a pore which can be broken into 2 regions. Now we need to talk about these regions. Okay. See, अगर आप यहाँ पे ध्यान दो, so we have with this voltage sensing domain, ये हमारा segment 4 है. Now TTX is your toxin binding site, यानी कि क्या, अगर कल को आप कोई drug use कर रहे हो, if it is binding over here, it is blocking your Na plus channel. If you're using some local anesthesia, it blocks in this region, जिसकी वजह से क्या होगा, आपका nerve impulse, pain nerve impulse जो होता है वो block हो जाता है, and because of which you don't feel pain. The signal is not given to the brain and hence you don't feel pain. Okay. अगर आप अपने बॉडी के हर एक पेन नर्व्स को ब्लॉक कर दोगे, so you will never feel any pain in your body. So just go back and read about the CPA अगर कभी टाइम मिले तो, ठीक है? Okay, let's come back over here. The ions are conducted through a pore which can be broken into 2 regions. The more external portion of the pore is formed by the P loop regions. Can you see the P loop region? A P loop कौन बनाता है? These segments जो आपके 5 and 6 के बीच में जो ये लूप आ रहे हैं, हर एक डोमेन का आपका 5 and 6 में जो लूप आ रहा है, that is your P regions. Okay, the region between the segment 5 and segment 6 of the 4 domains. This region is most narrow for the pore and is responsible for the ions selectivity. In future exam में अगर आता है like what is the selectivity filter in the Na plus channel, then you have to talk about the P loop which is formed by the segment 5 and segment 6 of all the 4 domains of the alpha subunit of your Na plus channel. Okay? Now, the inner portion of the pore is formed by the combined S5 and S6 segments of the 4 domains. The region linking domains 3 and 4 is also important for the channel. This region plugs the channel after prolonged activation, inactivating it. Okay, वो इतना जरूरी नहीं है. So इतना clear है. So what is the takeaway message about the P loop? Because that is acting as the selectivity filter for the ions. Okay. Now coming to the gating mechanism now. Gating. Since it is a voltage gated channels, right? So we have to see how it is happening. So there are 3 conformational state. मैंने हमेशा क्या बोला था? There is 2, close and open. But here people have considered the inactivated stage of your sodium channel as well. Okay. So before an action potential comes, the axonal membrane is at resting potential, that is minus 70 millivolt, right? And Na plus channels are there in deactivated state. यानी कि क्या? ना बंद है ना open है. They are just totally deactivated. It is not in use. Now, in response to increase in the membrane potential to about minus 55 millivolt. So when we talk about the action potential, तब आपको पता चलेगा that when action potential is changing, it is changing because of the K plus ions that time. ठीक है? तो क्या होगा, K plus भी positively charge है, जिसकी वजह से minus 70 जो है आपका less negative हो रहा है. So it is coming the minus 55. Okay, this will result in the activation gates opening of the positively charged Na plus ions. So what will happen? पहले तो K plus आया तो उसने minus 70 को minus 55 millivolt कर दिया. And as you know, when there is a change in the membrane potential, that results in the conformational change in the sodium channel. Correct? So that resulted in the opening of the sodium channel. Conformational change होना मतलब opening of the sodium channel. Now when it got open, Na plus ion was able to come. So what had happened? Now minus 70 से minus 55 से plus 30 हो गया है. ठीक है? Because इतना positive ions अंदर आ गया है. Now because the voltage across the membrane is initially negative, as its voltage increases to and past zero, now it is said to be depolarized. Okay? ये एक पॉइंट को आपको समझना है that from minus 70 when the membrane potential becomes 30 plus 30, it is said to be depolarized. अगर minus 70 से minus 90 चला जाता तो हम क्या बोलते हैं, hyperpolarized. Okay, now hyperpolarization means the signal is inhibited. Depolarization means signal is pass, यानी कि एक न्यूरॉन से दूसरे न्यूरॉन में हमने सिग्नल पास कर दिया है. और अगर minus 70 की जगह minus 90 हो जाता, यानी कि वो सेल ने सिग्नल वहीं पर ही इनहिबिट कर दिया है and it is not passing to the next neuron. Clear? Is this much clear? This is really very interesting also, कि कैसे आपका ब्रेन काम करता है, कैसे आपकी मेमोरी स्ट्रांग होगी. Yes, able to follow, guys?
Yes, ma'am.
Huh?
Yes, ma'am.
Yeah. Okay. Now coming to the potassium channels. So now let's talk about the potassium channel. So this is also very similar to your sodium channel. So of course it is widely distributed. So they form the potassium selective pores that span the cell membrane. Okay. Now when the K plus voltage gated channel gets open, membrane potential goes from minus 70 to more minus 90 millivolt. Now this is because the positive ion is moving outward, making the inside of the membrane more negative. Now guys, अभी आपको एक चीज यहाँ पे समझनी है. So just now I said that जब K plus अंदर आता है तब आपका minus 70 से minus 55 होता है, right? Now what you guys have to understand is that time, now when that minus 70 से minus 55 हो रहा है, तब आपका वहां पे Na plus K plus ATPase channel is working. ठीक है, which is totally different. हम यहाँ इसको बाद में पढ़ेंगे. Now if only your K plus channel is getting activated, okay, this is different and this is different. Okay, Na plus K plus ATPase is different. K plus channel, voltage gated channel is different. अब यहाँ पे क्या हो रहा है, this has got the opposite effect. ठीक है, Na plus के केस में क्या होता है, Na plus will try to come inside. From extra, it will come inside the cytoplasm. But when K plus channel gets activated, वो क्या करते हैं, cytoplasm से बाहर जाते हैं extracellular की तरफ. So what is happening? There is a loss of plus ions, correct? And that is making more negative the inside of the cytoplasm. Is this much is clear? Don't worry about this, because eventually we are going to talk about Na plus K plus ATPase, and then how exactly one neurons are transferring the impulse from one neuron to another, and when does the K plus channel act and when does these channel acts, we'll talk about it. Okay, अभी के लिए इतना याद रखो that Na plus channel जब open होता है तो बाहर से अंदर की तरफ आता है. ठीक है, K plus channel जब open होता है तो आपका अंदर से बाहर की तरफ जाता है. Okay, now if I ask you question why this is happening, what will be your answer? क्यों ऐसा हो रहा है कि एक अंदर आता है एक बाहर आता है? Why this difference is coming?
To maintain the potential. so that when
हाँ,
action
but
potential does
why
happen,
this thing is happening? ऐसा क्यों? It's very
actually
simple. हाँ, हाँ,
the
yes.
sodium potassium pump actually keeps the sodium out all the time and the potassium in all the time, so that when the action potential actually happens, depolarization can happen by
Yes.
changing the potential.
Yes, yes, that is also one point, but that comes later. It's absolutely correct, Shruti, because it is maintained in such a way from the starting. So if this is your cell, okay, the concentration of K plus here is 150 millimolar. Okay, and here the concentration of K plus is around 4 to 5 millimolar. तो क्या हो रहा है यहाँ पे? A gradient is formed. So जब भी आपका K plus channel open होगा, what will happen? Obviously from inside it will go outside, right? Similarly, when we have got Na plus, so Na plus का concentration अंदर रहता है आपका around 20 millimolar और बाहर आपका Na plus का concentration है around 145 millimolar. So if my Na plus voltage gated channel is getting open, so what will happen? Because of the gradient, बाहर से अंदर आएगा. And that's why in the case of K plus, from minus 70, it is becoming minus 90 millivolt. Whereas in the case of the Na plus channel getting open, minus 70 से ये minus 55 में जा रहा है. Now getting the point why this difference is coming? Yeah. Now we always need to make this difference. We always need to make this difference, right? As you said, the action potential तभी होगा, तभी आपका जो सिग्नल है वो पास होगा. तो जब आपका वोल्टेज गेटेड चैनल ओपन होता है, so it tries to reach equilibrium. But I don't want equilibrium to be reached. Otherwise, there won't be possible. It won't be possible for the cells to carry out the impulse conduction. तो उसके लिए हमारे पास है Na plus K plus ATPase. ठीक है. अब ये क्या करता है? K plus against the concentration gradient डालता है, यानी कि बाहर से अंदर की तरफ डालेगा. Na plus को क्या करेगा? अंदर से बाहर की तरफ डालेगा, लाइक अगेंस्ट द कॉन्सेंट्रेशन ग्रेडियंट. एंड इसलिए हमें यहाँ पे एटीपी लगता है. ठीक है. So Na plus K plus ATPase channel and Na plus channel K plus channel, they all work together when there is a action potential happening. Clear guys? पूरी कहानी?
Yes, ma'am.
Yeah, it will be more clear once we talk about the Na plus K plus ATPase channel also. Okay. So the structure. So now look at the structure over here. So it's a tetrameric structure. अभी हमने क्या देखा था? Na plus में क्या है? It has got the two subunit, alpha and beta, in which beta they have not mentioned. I didn't get the information about the beta subunit. What I found was only about the alpha subunit, which has got the 4 domains. Each domain is having 6 alpha helices. ठीक है? जिसमें से भी आपका fourth segment जो है, it is playing important role in selecting the ions. Now, in this case, in the case of the potassium channel, it's a tetrameric structure in which 4 identical protein subunits associate to form a complex structure, this one. Okay. Now, at the center, there is a cavity. In the cavity, these amino acids, okay, these are the loop of the helices, okay, which is actually forming the selectivity filter. SF stands for selectivity filter. So you can see over here, these amino acids are making interactions one at a time. Okay? So maybe in this cavity at a time, 3 can come. I don't know, the figure says like that. So if suppose 3 can come, it will make sure that these interactions are proper, then only it will get open up from here. ठीक है? तब जाके वो 3 आपके release करेगा. So the potassium selectivity filter, potassium ion channels remove the hydration shell from the ion when it is entering the selectivity filter. So selectivity filter is formed by 5 amino acid residues. Those are TV, GY, GG. So what are these amino acids? T stands for which amino acid?
Threonine.
Ha. Then V.
Valine.
Yes. G.
Lysine.
Correct. Y.
Tyrosine.
Tyrosine. Very good. Then again G is it, so glycine. Okay. So these are the amino acids, 5 amino acids, which is forming the selectivity filter. Okay. So the signature sequence is within the loop between the 4 helix. Now what was the 4 helix? If you have seen in the figure, agar notice kiya ho, these are your 4 helix. ये पोर हेलिक्स के जो लूप रहते हैं, those are interacting with the ions. This signature sequence is highly conserved with the exception that a valine residue in prokaryote's potassium channel is substituted with isoleucine in the case of the eukaryotic potassium channel. Okay. Now the four sets of electronegative carbonyl oxygen atoms are aligned towards the center of the filter pore. So what does it mean? ये कौन से electronegative carbonyl oxygen की बात कर रहे हैं? Can you tell me? So each peptide bond, can you see over here the amino acids they have mentioned which forms the selectivity filter? So unke oxygen atoms are actually protruding outside, which is making the interactions with the your ions. Okay? So the distance between the carbonyl oxygen and the K plus in the binding sites of the selectivity filter is same as between the water oxygens in the first hydration shell and the potassium ion, and hence they are able to form a energetically favorable route for desolvation of the ion. So iska kya matlab hua? As I said, it has to be so strong and in nearby distance so that K plus के ऊपर जहाँ पे जो water molecules आ रहे हैं, okay, so that are replaced, those oxygen are replaced by the oxygens of the amino acids. Okay? And then only those interactions are possible. Did you get the point here? How it is happening?
Yes, ma'am.
हाँ? श्रुति पूरा clear है? Okay? Yeah. So each time, okay, so it's not like only one K plus is coming at a time, but every K plus needs to get dissolved. यानी कि उसका hydration shell हटना चाहिए. That should be replaced by the oxygens of the selectivity filter when it is possible. Then only it is able to pass through the channel. ठीक है? So ऐसे ही करके सारे आपके K plus ions will be passed through the channel. Okay? So this was all about your potassium channel. Yeah, that's all for today's, guys. So maybe in the next lecture, we'll be talking about the ligand-gated channels.